Hello everyone, welcome. So in this video we're going to be looking at chapter 5a and we're going to be looking at photosynthesis and just the general process of photosynthesis. So biology is all about life, as we know, and we've learned about in Area Study 1, we've learned about the instructions for life, how we can use these functions to benefit science. Now what we're going to do in Area Study 2 is we're going to learn about the key processes that occur within a cell to keep it alive. So we're going to be looking at two processes. We're going to be looking at photosynthesis and cell respiration in both plants and animals, and we're going to be looking at how those processes create energy so that we can live. So we'll be starting with photosynthesis in chapter 5, and then chapter 6 we'll be moving on to cellular respiration. So just a disclaimer, so throughout chapter 5a we're going to be looking at photosynthesis, and we're going to be looking at it in its most general form. So there's a couple of different types of plants which we're going to go through in chapter 5, but just a disclaimer that the process that I'm about to go through with you for photosynthesis is for C3 plants. So C3 plants is basically your just normal, traditional, everyday plant that you'll just see out in the world. And this is most plants. And they have the, they just use the standard mechanisms of photosynthesis and they undergo typical photosynthesis. In 5b, we'll be learning about different kinds of plants and how how photosynthesis changes depending on the plant. But for what I'm doing today, I'm focusing on the majority C3 plants and just your typical photosynthesis pathway. So the first point, we need to be able to understand the general structure of the biochemical pathway in photosynthesis from reactants to product. So really luckily for you, when I was in school this was not the case, but luckily for you, they have said that you do not need to know any of the chemical symbols or anything like that. All you need to know is the inputs and the outputs, just in their word form. If you would like to know their chemical forms and their numbers and all that, absolutely go ahead. It's definitely not going to hurt it. It will probably help you, but you just need to know the inputs and the outputs. So photosynthesis, hopefully you've heard it before in year 10 and year 11. It's the biological process where photoautotrophs, so that's just things that feed themselves using light, they capture light energy from the sun and convert it into chemical energy. And when I say chemical energy, I mean glucose, sugars, and that's how they, how they provide the energy to function as a plant. This is obviously specific to plants. Animals do not do this. We do a different process, which we'll go through later on. So we've got this diagram, which is basically photosynthesis in a nutshell and what I would take away from this entire PowerPoint, honestly. So we have got our inputs on this side of the line, which is carbon dioxide, so 6 CO2s in particular, and water, so 12 H2Os. So they are the two inputs. So they're what's going to be going into this chemical reaction. We then have this arrow here that's signaling to our products, but please note that sunlight is on top of this arrow. So the reason that we put sunlight here is because it's not an input, it's not an output, but it does facilitate this entire process and is really important to include, but it doesn't get used up or created during the process. So we put sunlight here on top of the arrow, so it's really important to note that that's where it goes. We then have our products. So using carbon dioxide, water and sunlight, we then produce glucose. That's the chemical formula for glucose, carbons, hydrogens and oxygens. We produce some oxygen as well, and we produce some water as a product. So they are the inputs, they are the outputs. Let's go into a bit more detail about how that actually happens. So this is just a simplified diagram of where those inputs and outputs are coming from, and hopefully from your year 11 studies you will remember a lot of this. So our inputs, like I said, was carbon dioxide. Okay, so carbon dioxide is a gas. It's the gas that we breathe out. It is a output of cellular respiration, which we'll go through later on. So it's the gas that we breathe out that goes into the atmosphere, and our plants will absorb this or take this up into their leaves through their stomata, which is just little holes that allows gas exchanges. We also had our input of water. So our water is coming from our roots, okay? So our plants will be in the soil, hopefully they're being watered or rained on, and their water will be taken up via the roots into the leaf. So that's where our two inputs are coming from. We also obviously have sunlight, and you know where that comes from. It comes from the big yellow thing in the sky, which is also going to be a facilitator of this process. We then have our outputs. So our water and our carbon dioxide is coming in, and then we have our oxygen that's coming out. So during this process, we produce oxygen. So this also comes out of the stomata, and this is why plants are so important to us, because this oxygen that is a byproduct of their photosynthesis is what we breathe in. So we literally could not live without this product here. We also have a little bit more water as a product as well. So that water is probably going to be released from the stomata as well, unless the plant needs it, and just evaporate into the atmosphere. And then we've got our sugar. So our sugar, which is the main product of photosynthesis, is what is giving the plant the energy to perform all of its functions. So the sugar is probably going to go to the rest of the plant. It's going to provide that energy. Some of that sugar might be stored in the roots as starch and things like that, but that sugar is the most crucial part. So that's where our inputs and outputs are coming and going. So this is what we need to know. So we need to know the inputs, outputs, and locations of two different types of reactions that occur in photosynthesis. So light-dependent and light-independent stages, which we'll go through in C3 plants. So like I said, this is just in your typical plant. We'll go through our special plants later on. We also need to be able to understand the role of enzymes and coenzymes, which you hopefully remember from chapter 3, and how they help in facilitating photosynthesis as well. So I'll point those out as we go. So, really important before we get into all the processes to understand the structures that are involved in photosynthesis. These might be words you've heard of before, maybe not. Really good to have in your glossary so you know what I'm talking about when I'm talking about it. So, leaves are the main site of photosynthesis, which have a large surface area for light absorption. So, the reason that you see, really, long, thin, wide leaves, and the reason that your houseplants will always turn towards the sun is because leaves are the main site for this, and they are the site of our light absorption. So, they're really important. So we've got these cells, obviously leaves are organic things made up of cells, and mesophyll cells are the really specific cells that are inside leaves that undergo this photosynthesis process. So mesophyll cells are really important because they contain a large number of chloroplasts. So hopefully you remember from your organelle days that our chloroplasts are the main organelle that's responsible for photosynthesis. And these chloroplasts contain a pigment called chlorophyll, which is why plants are green. And this is what's going to capture the light energy. So our leaves are, made up of a bunch of mesophyll cells. Inside those cells are a bunch of chloroplast organelles, and inside those chloroplast organelles is a pigment called chlorophyll, which is going to facilitate our photosynthesis process. We also have these things called stomata, which I've talked about just previously, which are just tiny little holes in the leaves, and there's what's going to allow the gas exchange. So obviously we need carbon dioxide to be coming in, and we need oxygen to be going out, but they're also really important for water conservation as well. We've got our water that's absorbed through roots. We hopefully know what the roots of a plant are. They're normally in this part of the pot here. And we've also got our xylem. So xylem is, if you remember your xylem and your phloem, so xylem is the main transport system in which water travels around the plant after it's absorbed by the roots. So these are our key structures that I'm going to be talking about today. So make sure you've got those in your glossary. So we've got our chloroplasts, which I'm going to dive a little bit more deeply into. I'm just going to enter all of these there so we can see everything. So chloroplasts are the organelle that's inside our mesophyll cells. So this is a more, realistic representation, and this is your cartoony representation, but both will work. So a chloroplast is enclosed by an envelope consisting of two membranes. So that's important to know. So it's probably shown better in the cartoon. So it's consisting of two membranes on the outer side. The membranes are also divided into the interior of the chloroplast into compartments. Okay, so this is where all of our compartmentalization happens, which is really important because these compartments are the locations of photosynthesis, so we need to wrap our heads around this. So we've got a couple of different things. So we've got these thylakoid stacks. It's probably easier to see in the cartoon, so I'll go with the cartoon. So we've got these thylakoid stacks, and these thylakoid stacks are stacked into structures called grana. So we've got these little thylakoid discs, and all of those thylakoid discs that are stacked on top of each other are called the grana. So when I asked about the location of the light-dependent reaction, which we're going to learn about in a second, you can either say the thylakoid membranes or you can say the grana, and both would be correct. So this is where all of our light dependent stuff happens, in these thylakoid / grana here. We also have the stroma. So the stroma is all of the space or the fluid that floats around the thylakoid. So it's all of this, not empty space, but it's all of this space that is around the thylakoids or the grana stacks. So they're the two main locations that we're going to be looking at, and I just wanted to draw your attention to. Obviously, chloroplasts have other things too. They've got ribosomes and DNA, all of that sort of stuff, but they're the two main structures that we're going to be looking at for photosynthesis. So this is a really simple breakdown of photosynthesis and what it is. So as I said, it's split into two main stages. So it's split into a light-dependent stage and a light-independent stage. So hopefully the names give away the difference. The light-dependent stage requires light to work, and the light-independent stage does not require light to work. Okay, so like I said, the light-independent stage occurs in the thylakoid membranes or the grana. You can also use that as well inside the chloroplasts. The main enzyme that's involved in this process is ATP synthase, which we're going to look at in a second, look at what that does. We also have the light-independent stage. So this occurs in the stroma, so in all the space around the thylakoid membranes in the chloroplast. The main enzyme that's involved there is rubisco, which again we'll dive a little bit deeper into. So we've got obviously our two key enzymes involved in these processes, but of course do not forget that a range of coenzymes are working here as well, and we're going to be looking at those and how they help these processes. So let's go through this in more simplistic version, and then let's dive a little bit deeper. So we've got the energy from the sun. We know that that's important. Okay, so we've got our one of our reactants, one of our inputs, which is water. Okay, so water is used in the light-dependent stage, and then you'll see our other input, which is carbon dioxide, is used in the light-independent stage. So they're both inputs that is used at different stages. So we've got our two inputs here. We've got our sunlight and our water for the light-dependent phase. So this is where we're going to capture energy, sunlight energy, and we're going to split water. We're going to split this light energy into oxygen and hydrogen. Okay, so this is where we're going to use light energy to split water into oxygen and hydrogen, which we'll go into a little bit more detail. This is all these things that I'm talking about here, which we'll go through a bit later on. And then we've got our other input, which is carbon dioxide, which is going to go into the light-independent phase. And this is where something called carbon fixation happens. So this is where we take carbon dioxide and we turn it into an organic form that can be used to make glucose. So we take the carbon in carbon dioxide and we transform it into something that can be used as the carbon in glucose. So this whole fixation process produces water, and the main product is glucose. I forgot to mention as well that this splitting of water, the product there is oxygen. So let's dive a little bit deeper, because this is a very, very simplistic view. Let's go to the light-dependent stage. All right, so this is where we need light. This is where sunlight is really important. So in the first stage of photosynthesis, plants depend on light to split water. Okay, so this was our first input. Okay, so this is where that input comes into play. So what's going to happen is water is going to be inside of the chloroplast, and the light that's absorbed through the chlorophyll is going to split this water. So this process is called, it can be called two different things. You might see two different things. It could be called hydrolysis or photolysis, depending on what textbook you're reading, but those two words mean the splitting of water. And we know that hopefully we know that the chemical formula for water is H2O. So that water is being split into oxygen and hydrogen. Okay, so that's what it's being split into. So we know that this oxygen is a product. So that's what's going to leave eventually. It's going to leave the plant, and the hydrogen is going to be used a little bit later on. Okay, so that's the main purpose of the light-dependent stage. It's this hydrolysis, this splitting of water into these two main components. So the purpose of this first stage is to generate high-energy coenzymes. Okay, so remember coenzymes are things that facilitate or help enzymes. The coenzymes that are mostly used in photosynthesis are NADPH and ATP. So these coenzymes are really important because they power the second stage. All right, so they're made in the first stage, they're made in the light-dependent stage, so that they can be used in the second light-independent stage. So the main inputs of the light-dependent stage are, so water, we know. We know that from our main chemical reaction that we look at. But there's also these two things as well that are important not to forget. So water is obviously an input. It's split by light into oxygen and hydrogen. But these two inputs are really important. So these are the unloaded versions of our coenzymes that are going to be really important in our second stage. So you'll see that they're unloaded because they don't have the hydrogen there and they don't have that extra phosphate there yet. So they are unloaded. So NADP plus and ADP are the unloaded versions of these coenzymes. So they're going to be inputted into this process and through this process are going to become their loaded versions. So our outputs are oxygen, as we know from our chemical reactions. So that splitting of water is going to create oxygen that's going to be released eventually, but it's also going to create hydrogen. So that hydrogen that's created through that splitting of water is going to allow the addition of these hydrogen molecules into these coenzymes. So the unloaded versions go in and the loaded versions go out, and they're going to be used in the light independent reaction. Okay, so let's have a look at it in what looks like a complicated diagram, but actually is not. So we've got our lights obviously coming in. We've got water already in the chloroplast. That water is going to be split. It's going to be split into oxygen that's going to be released. It's going to be split into hydrogen. So you can see this hydrogen starts floating around, the thylakoid membranes inside the grana. Okay, so it's floating around. And you'll see that this ATP synthase enzyme here is cycling those hydrogens through, and that's going to do two things. So that's going to turn ADP into ATP, and it's going to turn NADP into NADPH. So this enzyme here, ATP synthase, is, as the name suggests, it synthesizes ATP. Okay, so what happens is those hydrogen ions that are released from the water, they power ATP synthase. So they pump through ATP synthase, and this powering allows us to turn ATP into ATP. So it allows us to add that molecule on there, and it also allows NADP to be transformed into an NADPH by including that H plus ion on there. So the H plus ions are really, really important for being added onto NADP to form NADPH, and it's also really important to power ATP synthase so that ADP can be transformed into ATP by the addition of a phosphate that would already be present inside the cell. Okay, so the main purpose is to create those coenzymes that are going to be really, really important for the next stage. So this NADPH and ATP are the really important parts of this stage. We then have our light independent stage. So this is a stage of photosynthesis that does not require light, obviously, as the name suggests. So instead, the reactions are energized by the coenzymes that we just made. So they're energized by ATP and NADPH coenzymes. So this occurs in the stroma, as we said, and is facilitated by enzymes. So this is the really enzyme-coenzyme driven part of photosynthesis. The first stage is like the preparations, and this is the real, the stage that produces all the really important stuff. So our inputs, we know that carbon dioxide is an input, so that is what's going to come through the stomata, and this is going to be used in something called carbon fixation. And we also have our inputs from the light dependent stage. So these have come from the stage we just learnt about. So we've got our now fully loaded NADPH with our H plus ion added, and we've got our fully loaded ATP with our phosphate added through ATP synthase. So we've got our fully loaded coenzymes ready to go. We then have our outputs. So by the end of this, we're going to have a fully functional glucose molecule, which is the main energy source for plants, and it's the main product of this photosynthesis. We're also going to have water as a just a byproduct of this, as another product. And then note that we're also going to go back to our uncycled versions of our coenzymes. So our coenzymes are going to be used up in the light-independent stage, and then they're going to go back to their unloaded form. So they're also going to be a product of this as well. So let's have a look at this. So our light independent stage, it goes through something called carbon fixation, which we're going to learn a bit more about in 5B, okay? But for now, it's just called carbon fixation. So the purpose of this is we've got our carbon dioxide input here. So it's going to go through this carbon fixation process. And the main reason for this process is to change inorganic CO2 into an organic version of carbon that can be used in glucose, okay? So the carbon that's inside of carbon dioxide, so the carbon here, cannot be used in this organic material. So what it has to do is it has to undergo a fixation process, which we'll learn the details of later, to turn itself into an organic carbon that can be used to create this glucose molecule. That's really, really important for the plant. This carbon fixation process heavily relies on the activities of NADPH and ATP. So they're our coenzymes that we loaded up in the first stage. So they are converted back into their unloaded forms because they're used up in this carbon fixation process. All right, and then we have our product, which is glucose, or our sugar molecule that the plant really needs. Something that's missing from this diagram is that another product is water. Okay, so during this process, during all these chemical reactions and things like that, water is also a product that's produced here that's in our chemical reaction. So that's the light independent stage. So a summary. So if you're going to take anything away from this, this is what you need to take away. You need to know the light independent stage. You need to know its location. So you need to know that it happens in that grana or those thylakoid membranes. You need to know what goes in. You need to know that water goes in. You need to know that sunlight facilitates and breaks this water down. You need to know that the unloaded forms of these coenzymes here also go in. You need to know that outputs are oxygen, which occurs from the splitting of water. And you need to know that this splitting of water also allows our coenzymes to be loaded with their H ions that came from the water, and also ATP can be loaded by the powering of ATP synthase as well. So you need to be able to understand the locations, the inputs and outputs, and the purpose of the light dependent stage. The purpose is really important. The purpose is to create these coenzymes that are going to be super important for our next stage. You then need to know the light independent stage. You need to know that it occurs in the stroma. You need to understand that an input is carbon dioxide, which comes in through the stomata. And you also need to understand that another two inputs are the coenzymes that were fully loaded in the stage beforehand. So our NADPH and our ATP. What happens is carbon dioxide undergoes carbon fixation, which allows it to be to form this glucose molecule. So the carbon in here gets transformed into organic carbon to create our sugar molecule. You also need to understand that our coenzymes are used up in this process. So the NADPH donates its hydrogen and the ATP donates its phosphate to power the process of carbon fixation. So their unloaded forms are going to be now the output because they've been used up. And then water is also an output as well. And this is exactly what's shown in this diagram here. Hopefully that helps. If you have any questions, bring them to class and I will see you there.